Hi everyone, welcome to AP Biology. This is Miss Clickparrot, and we are going to be going over some chemistry in this video, as well as some properties of water. Now this video is the first of two, so this is part one, so you want to watch this one before you go on to part two. During this lesson, we're going to be going over a few things, and you want to be able to make sure that you can answer these objectives by the end of the lesson. Remember, you can always go back and rewatch these videos as many times as you need to make sure that you're well prepared for the first day of class. The first objective we're going to be talking about a little bit about is the differences between ionic and covalent bonds and how those influence biological molecules. We're going to talk a little bit about why atoms bond together. Then we're going to talk about water's structure and why it's a polar molecule. And then we're going to finish up with a brief overview of pH and how to determine the pH of a solution. Now, I want you to take a moment and see what you can remember about ionic and covalent bonding. Now, bonding can be broken into three main categories. Last year in Regents Chemistry, we talked about ionic bonding, covalent bonding, and metallic bonding. Now, metallic bonding is not really going to play a role in AP Biology as much, so we're going to focus on ionic bonding and covalent bonding. So in ionic bonds, they form between a metal and a nonmetal. Now remember, metals have a tendency to lose electrons and non-metals have a tendency to gain electrons. So as this diagram is showing, when we form an ionic bond, we're going to transfer the electron or the valence electron from the metal to the nonmetal. Now why would molecules do this? Well, if you remember, atoms like to have a full valence shell in order to be stable. Our noble gases are the most stable atoms that we have. So they're all trying to get the noble gas configuration. Now for this metal, either it can gain 7 electrons or lose 1. So it would be simpler for it to lose the 1 valence electron. The nonmetal, however, could either lose 7 or gain 1 electron. So it would make more sense for it to gain. So by losing this valence electron to the nonmetal, the metal has a full valence shell. It'll lose this outer shell and then it'll just have the 2 valence electrons on the inside. When the nonmetal gains the valence electron, then it is going to complete its outer shell, making both of them more stable atoms. So when we do that, we're going to create a positive ion, which we call a cation for the metal, and then a negative ion or an anion for the other. The reason that these two are going to stay together is the electrostatic attraction between the cation and the anion. Remember, opposites attract. So once you make this anion and cation, they're going to be attracted for one another. Some examples of ionic compounds would be salt, common table salt, which is NaCl. Remember though, any salt any ionic compound is considered a salt. So magnesium chloride is also salt, and it is ionic as well. Covalent bonding, on the other hand, is between two or more nonmetals. So in this case, we have 2 fluorines that are bonding together. Now, why are they bonding together? Well, they're trying to fill their outer shell. Each fluorine on its own has 7 valence electrons. It shares 1 pair of electrons so that each atom has a full outer shell, making them both more stable. Now, covalent bonds can be equal or unequal depending on what elements we're dealing with, and we will talk about that in a moment. Some examples of compounds that are important to biology that have covalent bonds would be water. Water is a molecular substance with covalent bonding, as well as this molecule, which is a very important molecule in biology, C6H12O6, and it is glucose. Now, covalent bonding can get a little bit more complicated because it can be broken down even further. We can have multiple covalent bonds in covalent molecules. So you can have single bonds, you can have double bonds like in carbon dioxide, or you can even have triple bonds. So in bio, we're going to talk a lot about energy and energy being stored in bonds. So a single bond is going to be easier to break than a double bond, and a double bond would be easier to break than a triple bond. You can remember that if you picture yourself holding sticks. So say these are your 2 hands and you have 1 stick between them, you could probably break it pretty easily. If you held 2 sticks between your hands, it's going to be more challenging. It's going to be more difficult to break, and a triple bond would be even more difficult to break if you had 3 sticks between your hands. So you can kind of think about that. of the strength. So your triple bond out of the 3 is the strongest. We can also break down covalent bonding into nonpolar and polar covalent bonds. Polar covalent bonds are unequal sharing of your electrons. So this happens when one of your atoms has a higher electronegativity than the other atom. Whatever atom has the higher electronegativity is going to have a stronger attraction for those electrons. So the electrons are going to spend more time with the chlorine than the hydrogen in this example. Nonpolar covalent bonds are going to have equal sharing of the electrons. A lot of times this is seen between 2 of the same molecules. So for example, H2 or O2, all of our diatomics. Now, one very important polar molecule that we're going to talk quite a bit about is water. So take a moment and write down everything you can remember about the structure of water and why it is a polar molecule. So if you wrote down that water has a bent structure, you are correct. Remember, water has 1 oxygen connected to 2 hydrogens. It does have an asymmetrical shape, making it polar. And its shape is due to these 2 lone pairs of electrons on the top. Now, we said that it's polar, and we know that polar molecules have an asymmetrical charge distribution and also asymmetrical shape. Now, why is that? Well, oxygen has a higher electronegativity than our hydrogens, so it's going to have a stronger attraction for those electrons. So the shared electrons in the bond are going to spend more time with the oxygen than they are the hydrogen. So if those electrons are congregating more around the oxygen, what is that going to mean for the charge distribution of the molecule? We're going to see partial charges. The oxygen is going to take on a slightly negative charge, and the hydrogens will take on a slightly positive charge. Now, this is going to be very important, especially when you put multiple water molecules together. Now, just a few important things about water. Remember, it is essential for life. No life can happen without water. The body, any organism's body, is composed of over 2/3 water. Water is a polar molecule. And that's due, like we said, to the differences in electronegativity, and it has an asymmetrical molecular shape. And really important, we form hydrogen bonds. Now, hydrogen bonds, the word is a little bit misleading because hydrogen bonds aren't actually bonds. They're intermolecular forces, and they're actually a very strong intermolecular force. So why is it that we're seeing a force holding together water molecules between the oxygen of one molecule and the hydrogen of another? It comes back down to these partial charges. The oxygen we know has a partial negative charge. Our hydrogens have a partial positive charge. So what's going to happen is we're going to have an attraction between that negative and the positive, and it forms this intermolecular force. Now, hydrogen bonds, although on the weak side compared to bonds, they actually are the strongest intermolecular force out there. Now, before we end this video, we're going to go over a quick overview of acids and bases. Now, if you took Regents Chemistry last year, we did an entire unit on acids and bases. In AP Bio, we're going to keep it a lot more simple. You just need to know, you need to be able to compare different solutions for how many more times acidic they are. And you want to know some basics about acids and bases. So remember, our acids and base scale goes from 1 to 14. Over on this side, near a pH of 1, we're going to see our most acidic compounds, solutions. In the center, we're going to have a pH of 7, which will represent our neutral pHs. And over here, you'll have a pH of 14, which represents your bases. And this is a continuum. You can have different molecules in each category. An example of a acidic solution would be like lemon juice. Water is a great example of a neutral pH. And detergents and soaps would be an example of a pH of a higher basic pH. Now remember, pH is really measuring the hydrogen ion concentration. So which of these 3 solutions, a pH of 1, pH of 7, or a pH of 14, would have the highest hydrogen ion concentration? And that is correct. Your pH of 1 will have the highest ion concentration, and your pH of 14 would have the lowest hydrogen ion concentration. Now, what is the other ion that varies inversely with hydrogen? That would be hydroxide, OH. So over here, we have a very high hydrogen ion concentration, but we'd have a low hydroxide concentration or OH concentration. Over here, we wouldn't see many H pluses, but we would see a bunch of OH minuses. Now, the pH scale is based on a logarithmic scale. So it's usually expressed in exponents. So a pH of 1 would be expressed as 1 times 10 to the negative 1. And a pH of 7, 10 to the negative 7, and a pH of 14, 10 to the negative 14. Now, we said that the hydrogen ion concentration is going to be very high for our pH of 1. Remember, a negative 1, 10 to the negative 1 is actually a larger number than 10 to the negative 14, so don't let that number throw you off. Another thing you want to be able to do is compare different solutions. So, for example, if we wanted to figure out how many more times acidic our pH of 4 is than a pH of 6, we would look at our logarithmic scale. Now remember, each jump on this number line is going to represent a 10-fold difference in the hydrogen ion concentration. So if we start at a pH of 6 and we want to go to a pH of 4, we have to figure out how many jumps in our number line that would be. So first we would go from 6 to 5, that would be one jump, that would be a 10-time difference, and then from 5 to 4, and that would represent another 10 times difference. So to get your total amount that the solution is more acidic, you would multiply the two. So it'd be 10 times 10. So this solution, a pH of 4, would be 100 times more acidic than a pH of 6. Okay, try this one. How many more times acidic is a solution of a pH of 1 than a pH of 5? To answer this question, you would have to figure out how many jumps it is on a number line. So we'd start at a pH of 5. So we'd have to jump from 5 to 4, 4 to 3, 3 to 2, and then 2 to 1. So that would be 4 jumps. Each jump, remember, represents a 10 times difference. So we would multiply 10 4 times, and we would end up at 10,000, meaning a pH of 1 is 10,000 times more acidic than a pH of 5. Now, where does pH fit in with biology? And we talked a lot about it in chemistry, but we want to apply it now to biology and living organisms. So one place in your body where pH is incredibly important would be your blood. So your blood has its own pH, and that pH is a very specific pH. Now, I like this little chart because it kind of shows you how specific of a pH your blood needs to stay in order to keep you healthy. So your normal blood pH is somewhere between a 7.35 and a 7.34. It's a very small range between them. You're only looking at a 0.1 difference in pH. If you go just even a little bit lower, so let's say 0.4 lower, just a little bit under 7, we're looking at death. And if you go a little higher than that, you're also looking at death. So there's a very small range for which your pH needs to stay in your blood in order to maintain your health. So how does your body do that? Because your body's constantly producing carbon dioxide, which is acidic. So your blood would naturally get more acidic the more exercise you do. Well, within our blood, we have something called buffers. And buffers, basically what they do is they just help balance out that pH and maintain a constant pH level. So in your blood, for example, your blood will go back and forth, or will have a buffering system consisting of carbonic acid and bicarbonate. Basically what will happen if your blood gets too acidic, the extra hydrogens would be converted into carbonic acid, which would increase the pH of your blood a little bit, take some of those hydrogens out of the solution. If it gets too basic, your body will convert the carbonic acid into bicarbonate, releasing some of those H pluses. So we will talk about buffers a little bit later this year and how they affect biological systems and why they're important. But just keep in mind, right now, a buffer is just something that maintains a constant pH level. And your blood is a great example of somewhere that needs a buffering system. Now, during the first few days of school, we'll talk about these molecules. This is a protein. It's a 3D complicated structure, and proteins are going to be very influenced by pH. So as we talk more about this, I want you to kind of keep in the back of your mind that pH is going to play a large role in the formation of proteins, and hopefully soon you'll be able to tell me why. Okay, so make sure that you are able to answer all of these different objectives from this video. Remember, you can rewatch it and you can email us questions if you have any.